So moving on to number 5, this one starts with a pretty basic question. It says, researchers study mechanisms that enable or prevent speciation. A says, describe a post-zygotic mechanism that prevents gene flow and thus enables speciation. Okay, so this is from our final unit, and so post-zygotic would indicate that it is after the formation of the zygote. Now we know that for two things to be considered separate species, they need to be able to reproduce as well as have offspring that are both viable and fertile and all of that. So there are 3 post-zygotic mechanisms that you could mention. I'll put all 3 of them, but you only needed to put one. So the first one is going to be hybrid infertility. And you probably have to go on and give a definition of this. So this would just simply mean that the hybrids that are formed would not be fertile, and so therefore that stops them from being separate species. You would have hybrid inviability. So this would be when the hybrids are at a much lower fitness. So either they die in infancy, they die as an embryo, or they are born but they're not as fit for the environment. So we'll just put here that it would be lower fitness of the offspring. Can't reproduce would be infertility. And again, you only had to put one of these. And then the final one, hybrid breakdown. Now, hybrid breakdown is when they can do the F1 generation, but the F2 generation has the accumulation of too many recessive alleles, so they will begin to break down, and so it would also be lower fitness, but it would take a little bit longer over like multiple generations. Of F2, or you could say over generations. Okay, so that is A, probably just 1 mark, and you could have said any one of these. Now to move on, it says new genes can evolve from non-coding regions of DNA. It is not until certain regulatory elements are present in the DNA that a non-coding region becomes a new functional gene that encodes a protein. These regulatory elements include a promoter, a 5' untranslated region, or UTR, followed by a start codon, and a 3' UTR followed by a stop codon, seen in figure 1. So they're just giving you the basic structure of a functional gene. This is specific to this AG gene, but all coding genes will have this structure where they'll have the promoter, where you have the binding, the 5' UTR, your exons, your introns, the main bulk of it, which is your AG repeat sequence, and then you always have a 3' UTR. Okay, so let's go to the next part of the question. Zoom this in a little bit. This says, researchers study the evolution of the family of antifreeze glycoprotein AG encoding genes in Gattidae, a family of marine fish known as cods. When present in the fish, these glycoproteins reduce the freezing temperature of the fish. The researchers compared genomic sequences in 9 cod species and 1 non-cod fish species, B. brosme. They recorded the presence or absence of the elements of functional AG genes as well as AG-like sequences that are similar to a functional gene but have undergone mutation and do not contain all the elements required to enable protein production. And this is seen in figure 2. Okay, so figure 2, you have a phylogenetic tree showing the evolution of AG genes. Now, this might look a little bit overwhelming to students, so I'll take you through this. Here you have your phylogenetic tree. So it's just showing you the descendants. They give you the nodes where all of these sort of arise. And then they are going to indicate to you, which is going to go into our first question, that there was a period of freezing temperatures between 10 and 15 million years ago that you can consider as kind of like a disturbance. So there's a change in the environment at this point here. And then we saw rapid speciation after that. Here is looking at whether or not they contain the functional parts of the gene, including promoter, AG repeat sequences, and 3' UTR. Everything after this point would have 5' UTR and the AG-like repeats because they are after that node. If it has a negative sign like here, it means it doesn't have, in this case, the promoter. A positive sign would indicate that they do have it. They tell you the average water temperature of each fish as well. So you can see that these top ones usually live at about 5 to 10 degrees Celsius, while these bottom ones live at 1.2 to 6 degrees Celsius, so a little bit colder. And then finally, you have this L and this P here. If there's an L beside them, like these ones, it means that they had the gene for the glycoproteins and then they lost them. And it tells you in the question that it was a similar gene but had undergone mutation. And then the P, the ones beside these ones, indicate that it's still a functional gene that's capable of making protein. Okay, so for part B, let's do this one. Part B says, based on the data in figure 2, explain how changes to the genome enable cods to survive and reproduce after a freezing temperatures between 10 and 15 million years ago. Okay, so this one's actually pretty straightforward, and it's not really asking you anything about this diagram here. Just think about it in general when you're changing the genome sequence. So the change of the genome sequence is going to change your phenotype. Okay, and after this period of freezing temperatures, you had a change in the environment, so the temperatures would have dropped. And as a result, the ones that had the mutation that had this antifreeze glycoprotein, because having the antifreeze glycoprotein means that you can survive at colder temperatures. So the ones that did have all of these functional genes were able to survive and would have been, selected for in this colder environment. The colder environment would have selected against these ones over here that don't have the antifreeze proteins. Okay. So it really comes down to natural selection and the fact that having the glycoproteins helps you be better equipped and selected for in this period right here where it did get colder. So for this one, we'll put that the genetic sequence, so it's asking specifically how the genome enables cods to survive. So we'll put genome changes / mutations. We'll say that it reflects the phenotype. So reflects phenotype. So those with the AG genes, which are for the glycoproteins, they would produce antifreeze proteins. And these ones would be selected for by the environment. By the colder environment, so during that period of freezing temperatures. And then you would also probably have to indicate that it would be vice versa. So reproduce after, explain how the changes to the genome enables cods to survive and reproduce after a period of freezing temperatures. Okay, so actually you wouldn't, because they're really just referring to the ones that survived after this. These ones over here did not have the antifreeze proteins, you can see, because these ones are missing all of the elements of the gene, and then these ones mutated to be non-functional. So everything within this 5 to 10 degrees average water temperature do not make those glycoproteins, therefore they're not equipped, or they're not fit to live at 1.2 to 2 degrees Celsius. However, they would have to live at a warmer temperature because that's the only way that they can survive. So we can just put species without, and we can say functional. AG wouldn't survive. So it's just a process of natural selection. Okay, so there's probably a bunch of different ways in which you can say this, but as long as you kept the idea of that the genome is going to change the ability to produce or not produce antifreeze proteins, and that is going to determine what temperature they can live at because of natural selection. So as long as I think you include those key points, you should be fine. Okay, let's zoom in on C here, which says, using the template in the space provided for your response, place an X on the phylogenetic tree to represent the origin of the functional AG gene. Now, I saw a couple students' comments. There was a little bit of disagreement on the answer for this one, so I'll put what I personally think the answer is. Again, we need to wait till the answer guide comes out, but it's asking you to represent the origin of the functional AG gene. Okay. So what's happening here is you're having the emergence of this AG-like gene right here. Okay, and then if we're looking, so these ones just completely are missing, all of the functional components of it. So these are not going to include any of the AG functional genes. Which ones do have the AG functional genes, though, are these ones over here. Now, the issue for students here is these two over here. So obviously these ones have functional AG genes. They have this P here, which means that they're functional. However, it's asking for the origin of the initial functional gene. And what they had mentioned in the question happened to these ones that have the L beside them is that AG-like sequences that are similar to a functional gene but have undergone mutation and do not contain all the elements required. So I believe these ones that have the L had functional genes, but they had lost them due to mutation. Okay, so you would still be able to include them in, having the functional gene, but they evolved after the emergence of that gene and they had a mutation that made it non-functional. So I saw a lot of students wanting to put it right here because these are the only functional ones. However, in my personal opinion, I think you would put the X right here because what had happened is you had the emergence of this gene being functional right here. And then what happened is it remained functional going this way, but it did not remain functional going this way. So the gene was functional at this point, and then you had it branch off to ones that maintain their functionality, and then the ones that mutated to not become functional anymore. I think that's kind of the key to the question, is that the genes were functional and then became non-functional, these ones with the L. So then you would have to put the X prior to that. You wouldn't be able to put the X right here because that would indicate that, this one has a functional AG gene, but it's missing all the components. You couldn't put it here because, again, you're indicating that all these ones have the functional genes, but they don't even have the components for it. So my final answer for this one is that you have to look for the common ancestor of all of these. Definitely the P's because they have, the present functional gene, and then I do believe the L's as well because they had a functional gene and then was then lost. So the origin of it when it originally, came about would be right here. So I would put my X right there. Okay, the final part, D, says, based on figure 2, explain how genetic differences among the species in the Gattidae family determine the habitats in which they can survive. Okay, so this one's a little bit of a strange question because it's pretty much the same as B, so I'd be curious to see the answer guides for this one, but it's the same type of thing. They're asking you changes in the genome versus genetic differences. So for these ones, I believe they specifically want you to address both of the different groups, which is probably the only difference between these ones. So I would go with this pretty much the same thing that I mentioned in part D, is that the genetic differences, so whether or not they have the AG-like gene, is going to determine what temperature they can live at because of they're always going to live where they're best fit. So for this one, I would say that those that do have functional AG gene they would produce antifreeze proteins and then be best fit or best suited, or you could also say selected for, best fit for or selected for in colder environments. I would make sure to mention the actual numbers. So I would mention in the colder environments 1.2 to 6 degrees Celsius. And then I would say that the ones that don't have the functional gene would not be able to survive in that temperature. Therefore, they have to live at the warmer temperatures because that's what they're most fit to live at. They're in no way fit to live at these colder temperatures because they don't produce those specific proteins. So non-functional AG, they don't produce the proteins. So we're not fit for the colder temperatures. And therefore they must live at warmer temperatures. And then you can include the actual metric here, so 5 to 10 degrees Celsius. Okay, so I think for both B and D, as long as you mention, the fact that it reflects the phenotype and talk about how this is reflecting the ability to actually produce these special proteins, and the environment is going to select for or against the fish species based on whether or not they can produce these proteins. Okay, if they can, the colder environment will select for them, and if they can't, the warmer environment will select for them. Okay, so that's the end of 5.